Hey guys, let's take a look at the cell. So before we kind of jump into the questions and everything that we want to cover in this video, the nature of what we'll go over here is, kind of like basic definitions of really all the parts of the cell. So there's a decent chance that you were already familiar with this before you took AP Bio, um, but nevertheless it is on the test, and so it's important that we go over each of these. So there are, a lot of slides on this video, but they really are all pretty quick. So anyways, we've got 5 questions here, and these questions will show up again at the end of the video, at which time you can sort of test yourself on it. What is selectively permeable and regulates which molecules are allowed to enter and exit the cell? Difference between smooth and rough ER. What converts oxygen and nutrients into ATP? What is the difference between active and passive transport? Sodium-potassium pump, what does that do? Okay, so those are the types of questions that you want to be familiar with. Okay, so we'll jump right into it at the very beginning with eukaryotes and prokaryotes. Eukaryotes have internal membranes while prokaryotes do not. Eukaryotes can be unicellular or multicellular organisms. A eukaryote is any organism whose cells have a cell nucleus and other organelles enclosed within membranes. Eukaryotes evolved about 1 billion years ago, while prokaryotes evolved 3.6 billion years ago. A prokaryote is a unicellular organism that lacks a membrane-bound nucleus, mitochondria, or any other membrane-bound organelle. Prokaryotes are divided into 2 domains, archaea and bacteria. The first living organisms were prokaryotes. So again, that's a lot of stuff that I would imagine you're already pretty familiar with. So we'll just keep going. So a lot of times in videos like this, or when you go over these things with students, is they know quite a bit of the stuff, but there are little knowledge gaps of stuff they've just forgotten. The nucleus contains most of the cell's genetic material, organized as DNA molecules, combined with a large variety of proteins to form chromosomes. The genes within these chromosomes are the cell's nuclear genome and are structured in such a way to promote cell function. The nucleus maintains the integrity of genes and controls the activities of the cell by regulating gene expression. All cells are surrounded by a plasma membrane. The membrane is composed of a phospholipid bilayer with proteins dispersed throughout the layer. The membrane is also covered in places with cholesterol molecules and proteins. The plasma membrane is selectively permeable and regulates which molecules are allowed to enter and exit the cell. The surface area of the plasma membrane, which has control over what enters and leaves the cell, limits the overall size of the cell. The reason for this is that the volume, 4/3 pi r to the third, increases at a faster rate than surface area, which is just 4 pi r squared. Therefore, the cell volume increases to the cube of the linear increase, while the cell surface area increases only to the square of the linear increase. In animal cells, the plasma membrane is the only barrier separating cell contents from the environment, but in plants and bacteria, the plasma membrane is also surrounded by a rigid cell wall, which we'll talk about in a second. Bacterial cell walls are composed of peptidoglycan. Yeast cells are composed of 2 layers of B-glucan, the inner layer being insoluble to alkaline conditions. Both of these are surrounded by an outer glycoprotein layer rich in carbohydrate mannan. Plant cell walls consist of multiple layers of cellulose. These types of extracellular barriers confer shape and rigidity to the cells. The cell wall gives the plant its actual shape. It acts as a gatekeeper because it determines what can come in and out of the cell in order to keep the cell protected. The cell wall protects the plant and cells from the many insects and pathogens that could harm the plant. There are holes all over the cell wall called plasmodesmata, These are holes that allow for nutrients to enter the cell as well as waste to exit the cell. Plant cell walls are primarily made of cellulose, which is the most abundant macromolecule on Earth. Cellulose fibers are long, linear polymers of hundreds of glucose molecules. These fibers aggregate into bundles of about 40, which are called microfibrils. Ribosomes are both synthesized and produced in the nucleus. The ribosome is the site of protein synthesis. Ribosomes link amino acids together in the order specified by messenger RNA molecules. Ribosomes consist of two major components: the small ribosomal subunit, which reads the RNA, and the large subunit, which joins amino acids to form a polypeptide chain. Each subunit is composed of one or more ribosomal RNA molecules and a variety of ribosomal proteins. The Golgi apparatus receives proteins and lipids from the rough ER. It modifies some of them and sorts, concentrates, and packs them into sealed droplets called vesicles. The Golgi apparatus is located near the nucleus and has several flattened membranous sacs. Smooth ER is devoted almost exclusively to the manufacture of lipids, and in some cases to the metabolism of them and associated products. In liver cells, for example, smooth ER enables glycogen that is stored as granules on the external surface of smooth ER to be broken down to glucose. Smooth ER is also involved in the production of steroid hormones in the adrenal cortex and endocrine glands. Large amounts of smooth ER are found in liver cells, where one of its main functions is to detoxify products of natural metabolism and to endeavor to detoxify overloads of ethanol derived from excess alcoholic drinking, as well as barbiturates from drug overdose. To assist with this, smooth ER can double its surface area within a few days, returning to its normal size when the assault has subsided. It is called rough ER because it is studded on its outer surface with ribosomes. These are called membrane-bound ribosomes and are firmly attached to the outer cytosolic side of the ER. Rough ER is found throughout the cell, but the density is higher near the nucleus and Golgi apparatus. Mitochondria are rod-shaped organelles that can be considered the power generators of the cell, converting oxygen and nutrients into ATP. ATP is the chemical energy currency of the cell that powers the cell's metabolic activities. This process is called aerobic respiration and is the reason animals breathe oxygen. The main role of chloroplasts is to conduct photosynthesis, where the photosynthetic pigment chlorophyll captures the energy from sunlight and converts it and stores it in the energy storage molecules of ATP and NADPH while freeing oxygen from water. The mitochondria produce ATP during aerobic respiration. Lysosomes break down cellular waste products, fats, carbohydrates, proteins, and other macromolecules into simple compounds, which are then transferred back into the cytoplasm as new cell-building materials. To accomplish the tasks associated with digestion, the lysosomes utilize about 40 different types of hydrolytic enzymes, all of which are manufactured in the ER and modified in the Golgi apparatus. So we have both active and passive transport. Active transport moves ions from a low to high concentration, uses a sodium-potassium pump, and requires ATP. Passive transport, on the other hand, moves ions from a high to low concentration and does not require ATP. Both active and passive transport use ion channels to move ions across the cell membrane and either into or out of a cell. The sodium-potassium pump functions to pump sodium ions out of the cell and potassium ions into the cell. The sodium-potassium pump is a well-understood example of active transport. Sodium and potassium ions are pumped in opposite directions across the membrane, building up a chemical and electrical gradient for each. These gradients can be used to drive other transport processes. So we're going to look, it just says isotonic on the screen, but we also look here at hypertonic and hypotonic and be able to compare and differentiate the three. Hypertonic means higher concentration of solute. In a hypertonic solution, cells shrink due to water flowing from a higher water potential to a lower water potential. Cell shrinking is referred to as plasmolysis. In a hypotonic solution, cells either swell or burst. A hypotonic solution is one in which the concentration of solutes is greater inside the cell than outside the cell. And a hypertonic solution is one where the concentration of solutes is greater outside the cell than inside it. Water flows from hypotonic to hypertonic solutions. In an isotonic solution, the concentration of the solute of the solutions inside and outside the cells are equal. Endocytosis is a form of active transport in which a cell transports molecules, such as proteins, into the cell by engulfing them in an energy-using process. Endocytosis and its counterpart, exocytosis, are used by all cells because most chemical substances important to them are large polar molecules that cannot pass through the plasma or cell membrane by passive means. Endocytosis includes pinocytosis, which means cell drinking, and phagocytosis, which means cell eating. So we'll look at both of those here. As we just said, pinocytosis is sometimes referred to as cell drinking. Pinocytosis is a mode of endocytosis in which small particles are brought out to the mitochondria and then suspend within a small vesicle. These vesicles subsequently fuse with lysosomes to hydrolyze or break down the particles. This process requires energy in the form of ATP. Phagocytosis is the process by which a cell engulfs a solid particle to form an internal compartment known as a phagosome. It is distinct from other forms of endocytosis, such as pinocytosis, that involve the internalization of extracellular liquids. Phagocytosis is involved in the acquisition of nutrients for some cells. In multicellular animals, the process has been adapted to eliminate debris and pathogens. Gap junctions, gap junctions are specialized intercellular connections between cells in most animals. Gap junctions are an example of cell-to-cell communication. They directly connect the cytoplasm of two cells, which allows various molecules, ions, and electrical impulses to directly pass through a regulated gate between cells. Gap junctions occur in virtually all tissues of the body, with the exception of adult, fully developed skeletal muscle and mobile cell types, such as sperm. Gap junctions, however, are not found in simpler organisms, such as sponges. Apoptosis is a process of programmed cell death that occurs in multicellular organisms. Biochemical events lead to characteristic cell changes and death. Triggering signals can come from the nucleus, the ER from outside the cell, and the mitochondria can initiate apoptosis by sending a relay protein to the cytoplasm. It's estimated that between 50 and 70 billion cells die each day due to apoptosis in the average human adult. In contrast to necrosis, which is a form of traumatic cell death that results from acute cellular injury, apoptosis is a highly regulated and controlled process that leads to advantages during an organism's life. Peroxisomes are catabolic organelles found in the cytoplasm of cells. The reason they're abundant in liver and kidney cells is because these cells have to carry out a lot of detoxification. They do it using an enzyme called catalase, which is present in peroxisomes. Primarily, the cytoskeleton gives the cell its shape and mechanical resistance to deformation. Through association with extracellular connective tissue and other cells, it stabilizes entire tissues. The cytoskeleton is involved in many cell signaling pathways. In the uptake of extracellular material, endocytosis, it segregates chromosomes during cellular division, and it's involved in cytokinesis, the division of a mother cell into two daughter cells. It also forms specialized structures such as flagella and cilia, which we'll talk about right now. Cilia and flagella move liquid past the surface of the cell. For single cells, such as sperm, this enables them to swim. They consist of a cylindrical arrangement of 9 microtubules. A pair of single microtubules run through the center of the bundle, producing the 9+2 arrangement. Cilia are short and flagella are long. Osmosis is the spontaneous net movement of solvent molecules through a semi-permeable membrane into a region of higher solute concentration. It may also be used to describe a physical process in which any solvent moves across a semi-permeable membrane. Simple and facilitated diffusion. Simple diffusion is the process by which solutes are moved along a concentration gradient in a solution or across a semi-permeable membrane. Simple diffusion is carried out by the actions of hydrogen bonds forming between water molecules and solutes. Water molecules move into surround individual solute molecules, which in turn maximize hydrogen bonding. Facilitated diffusion is a form of facilitated transport involving the passive movement of molecules along their concentration gradient, guided by the presence of another molecule, usually an integral membrane protein, forming a pore or channel. Facilitated diffusion does not directly involve high-energy molecules like ATP or GTP, since the molecules are moving along their concentration gradient. Simple diffusion requires no membrane. Facilitated diffusion involves molecules that diffuse through membrane channels. Water potential is typically expressed in potential energy per unit volume. With no restriction on flow, water will move from the locus of greater potential, which is pure water, to the locus of lesser, which would be the solution. Flow proceeds until the difference in potential is equalized or balanced by another water potential factor, such as pressure or elevation. And then finally, endosymbiosis. When one organism actually lives inside the other, it's called endosymbiosis. The endosymbiotic theory describes how a large host cell and ingested bacteria could easily become dependent on one another for survival, resulting in a permanent relationship. Over millions of years of evolution, mitochondria and chloroplasts have become more specialized, and today they cannot live outside the cell. Essentially, the theory of endosymbiosis states that originally chloroplasts and mitochondria were prokaryotes, and they resided in larger cells and formed a permanent relationship. Okay, and then if you're still with me, we have these questions that we started off with. So this is a great time to go ahead and hit that pause button and see if you can answer these questions for yourself.